wakefulness, attention, reward, stress, locomotion. Different behavioral states that exert powerful control over how the brain processes information. Such regulation of the brain function is mediated by neuromodulators such as dopamine and norepinephrine. Defections in the neuromodulatory pathways are associated with a number of neurological disorders and neurodegenerative diseases, such as Parkinson's disease, schizophrenia, and post-traumatic stress disorder. Neuromodulation acts through intracellular signaling pathways to control neuronal communication efficacy and plasticity. For example, binding of norepinephrine to beta-adrenergic receptors triggers a signaling pathway that activates cyclic AMP-dependent kinase, also called pKa. pKa, in turn, can phosphorylate various substrates to control synaptic transmission, neuronal excitability, and plasticity. Our approach is based on a sensor called A kinase activity reporter, abbreviated as ACAR. ACAR is a substrate of pKa. When ACAR is phosphorylated by pKa, its conformation will change from an open state to a closed state, pulling two fluorophores at two ends of the sensor close to one another. This changes the type of interaction between the two fluorophores into a fluorescence resonance energy transfer, or FRET, which can be measured using microscopy. The type of microscopy that we use to image ACAR is 2-photon fluorescence lifetime imaging microscopy, also called 2P FLIM. 2P FLIM can be integrated into conventional 2-photon microscopes by adding a defined number of hardware components. 2P FLIM has advantages in imaging FRET-based sensors in vivo, as it provides highly sensitive yet stable measurements, independent of the depth of neurons in the brain. We established a characterization pipeline in which we examined the sensor's response to norepinephrine in neurons. Based on this characterization, we improved the current best ACAR sensor by about 3-fold. We call this new sensor T-ACAR Alpha. We found that pKa activity is dynamically regulated during behavioral states. For example, when the animal is awake, there is a tonic level of pKa activity in most cortical neurons that are mediated by norepinephrine. When the animal runs, pKa activities increase in a fraction of neurons in the motor and visual cortices, but less so in the barrel cortex.